for any cellular mechanism to take place, the cell should have the ability of transport across its membrane. So the transport across cell membrane occur by various mechanisms. So the mechanisms can be classified as active transport, passive transport and vesicular transport. Now, active transport can be further classified into primary active transport and secondary active transport. Then passive transport, we have simple diffusion, facilitated diffusion and osmosis. And in vesicular transport, we have endocytosis, exocytosis and transcytosis. So let's see a little bit in detail about active transport today. Okay, so coming to the transport process, just a comparison between the active transport and passive transport. So let's take the analogy of a swing. So whenever you sit on a swing, just by the momentum of it, it starts swinging back and forth. It really doesn't take any effort. But if you really want to soar into the sky, someone has to give you a push from behind. So the passive swinging, okay, or the passive movement that you get simply while sitting on the swing can be compared to that of a passive transport mechanism, while the active push is required for the active transport mechanism, which actually requires expenditure of energy. The energy of the push is required to make the swing forward. Similarly, for active transport, energy is required to move the molecule from one side of the membrane to the other side of the membrane. So, active transport, like you've already seen, it's like an uphill movement of a particular substance. Okay, it's like rolling a stone up the hill, for example. So, here, to make it more clear, you can see that on the intracellular side of the membrane, there is one little red molecule over there, while on the extracellular side, you have many such red molecules. So, by all known laws of physics, the movement of the red molecule should be from the area of higher concentration to the region of lower concentration. But here, in active transport, what is happening is the cell is actually expending energy or breaking down ATP molecules to provide the energy for transport of the particular object from a region of lower concentration to a region of higher concentration. So under active transport, it is again divided into two types depending upon the source of energy that is being utilized. Okay, so we have the first one, that is the primary active transport, and the second one where you have the secondary active transport. In primary active transport, the mechanism of transport is actually occurring by direct hydrolysis of ATP, and we use the common example of sodium potassium ATPase to actually describe it. We also call it a sodium potassium pump, and the other primary active transport pumps that we have are the calcium pump and hydrogen potassium pump. These are the common examples that we use. Now, secondary active transport means there we are using the energy produced by primary active transport to create a gradient of a particular ion, and the transport occurs according to this gradient. Now, that is a topic for another session. So, here let's see a little bit in detail about the sodium potassium pump or sodium potassium ATPase. So, we will be discussing this particular topic under the following headings, that is the structure, mechanism, functions, regulation and applied physiology. So, this is the structure of the sodium potassium ATPase. The sodium potassium ATPase is basically a heterodimer, that is, it has two subunits, the alpha and beta subunits. The alpha subunit is the subunit through which the transport of the ions take place. Like the name suggests, the ions that are going to be transported are sodium and potassium, while the beta subunit is actually a mechanism for anchoring the entire protein across the cell membrane. Now, as you can see, this particular transport protein or the sodium potassium ATPase is a particular protein that transposes itself across the entire cell membrane. It is there crossing from the extracellular surface through the membrane into the intracellular surface. So, we have already seen it is a heterodimer with alpha and beta subunits. It causes transport of sodium and potassium, which occurs through the alpha subunit. The alpha subunit has two particular activities, that is the ATPase activity, and it has binding sites on the intracellular and extracellular surfaces. So let's see what those are. So on the intracellular side, you have site 1, which is the site for binding sodium ions. Then you have site 4, which is the phosphorylation site, and site 5, which is the site for binding of ATP. The three sites: sodium binding site, phosphorylation site, and ATP binding site. While on the extracellular membrane, you have two sites, that is the site for binding potassium and the site for binding glycosides, or for an example that is given over here is the orbine, so we call it as the orbine binding site. So now that we have learned about the structure of the sodium potassium ATPase, let's learn how the pump works. So here, as we have already mentioned, you have three sites on the inner surface of the alpha subunit, and on the outer surface you have the two other subunits. So on one side, on the intracellular side, you have the sodium ions, and on the extracellular side you have the potassium ion. So here the transport of sodium ion actually occurs against the concentration gradient. As you have already learned in the body fluid compartments, you know that the sodium content in the extracellular fluid is much more than the sodium content in the intracellular fluid. So a significant amount of energy will be required for the transport of sodium from the intracellular side of the membrane to the extracellular side. So where do we get the energy for that? We get the energy from ATP. Now the ATP will come and bind to the ATP binding site, where it has the ability to break down the ATP to ADP and inorganic phosphate. Now this phosphate will go and bind to the phosphorylation site, and it causes a conformational change of the alpha subunit. Now meanwhile, the sodium ions have already bound itself to the sodium binding site, and as the conformational change occurs, the sodium ions, that is 3 sodium ions, will be pushed outwards because of the conformational change in the alpha subunit. Okay, so that is how sodium goes from the inner surface of the membrane to the extracellular fluid. Now from the extracellular fluid, we have to transport potassium inside. Okay, so what happens there? When potassium comes and binds to the potassium binding site, there it generates another conformational change, reverting the structure back to the original state, and you have the potassium ions that are flowing back inside. Okay, so it is simply a conformational change that is generated in the alpha subunit because of the breakdown of ATP that is helping with the transport of ions across the membrane. So in a nutshell, let's see the mechanism once again. The binding of sodium and ATP on the intracellular sites will transfer phosphate from the ATP to the phosphorylation site due to the ATPase activity of the alpha subunit. It results in a conformational change in the alpha subunit, and it transports 3 ions of sodium to the extracellular fluid, while 2 ions of potassium will bind itself to the alpha subunit on the extracellular binding site, and it causes dephosphorylation of the phosphate binding site and changes the conformation of the alpha subunit once again, and the 2 potassium ions will be flowing into the intracellular fluid. So where is our sodium potassium pump found? It is basically found in almost every cell that is present in our body, and its peculiarity is that it is an electrogenic pump. You've already seen that it transports 3 ions of sodium to the extracellular fluid and 2 ions of sodium into the intracellular fluid. So basically it is creating a net electronegative charge inside the membrane, and outside the membrane becomes electropositive because of the excess number of sodium ions that is going. So because of that, we say that the sodium potassium pump has a coupling ratio of 3:2. That is 3 ions of sodium moving outside and 2 ions of potassium which is moving inside. Okay. So the sodium potassium pump, as we have already seen, it is present in all the eukaryotic cells, and it functions are that it maintains the sodium-potassium concentration difference across the cell membrane, it helps in maintenance of the cell volume, and it causes electronegative charge inside the cell. That therefore it is called as an electrogenic pump. And how is the activity of the sodium-potassium pump regulated? So you can have an upregulation or a downregulation of the activity of the sodium-potassium pump. You have various chemicals or substances that will facilitate with that. So if there is an increase in the intracellular sodium level, or if the thyroid hormone, aldosterone and insulin, all these substances can cause an increase in the activity of the sodium-potassium pump, while a decrease in the activity of the sodium-potassium pump is brought about by glycosides like ourbane or dopamine. Okay, so these are the factors that can cause regulation of the sodium-potassium pump. So coming to the most important aspect that we get to see, anything that you learn in physiology has its applications in the clinical level. So here the sodium-potassium pump is the main target in treatment of cardiac failure. See, there is a drug called as digoxin. So this digoxin is a cardiac glycoside which actually binds to the ourbane binding site that we saw earlier. It binds to the ourbane binding site, and what it does is it will inhibit the sodium-potassium pump. So coming to the clinical aspects of the sodium-potassium pump. So anything that you learn in physiology has its application at the clinical level. So we are going to learn what is the application of sodium-potassium pump. So in treatment of cardiac failure, we use a drug called digoxin, which is a cardiac glycoside. Now this cardiac glycoside, it will bind to the glycoside binding site, or we call it as the ourbane binding site that we saw earlier. So when it binds to the ourbane binding site, what it does is it causes inhibition of the sodium-potassium pump. So the sodium remains inside the cell and the potassium remains outside the cell. So that pump stops working. So what happens is this energy that was generated by the sodium-potassium pump will be used in the cardiac cells for the exit of calcium. Okay, that excessive accumulation of calcium should be prevented in a cardiac cell under normal conditions. So for the removal of calcium, we actually require the energy that is being provided by the sodium-potassium ATPase. But in cardiac failure, what is happening is the cardiac muscle cells have an inability to pump. So here what we are doing is we are giving them an extra push with the we are facilitating the release or retention of more calcium within the cell, which will actually help the cardiac muscle cells to pump more blood to the various parts of the body. So here what we are doing is we are helping in retention of calcium. So the sodium-calcium exchanger will cease to work without the action of the sodium-potassium ATPase, and the calcium will remain inside the cell. So when calcium remains inside the cell, it is a potent stimulus for cardiac muscles to contract. It actually increases the myocardial contractility. Okay, the ability of the myocardial cells to contract. So this is one mechanism by which we can treat cardiac failure. So one of the drugs that we use is digoxin, and digoxin actually acts by inhibiting the sodium-potassium pump. Okay, so again the same thing, that is cardioglycosides like digitalis is used in management of heart failure and inhibits the sodium-potassium pump. It helps in accumulation of sodium inside the cell and prevents the potassium influx. The intracellular accumulation of sodium decreases the sodium gradient from exterior to the interior of the cell, and because of that, the calcium efflux through the sodium-calcium exchanger in the membrane utilizes the sodium, which actually utilizes sodium gradient, is disabled, and decreased sodium gradient will cause a decrease in the efflux of calcium, and it promotes an increase in the intracellular calcium concentration and enhances the myocardial contractility. So to summarize, sodium-potassium pump is situated in almost all tissues of our body. It is an electrogenic pump with a coupling ratio of 3:2. It has two subunits, the alpha and beta subunit. The alpha subunit is the functional subunit with 5 major binding sites. Then we went through the mechanism of the sodium-potassium pump, and finally the clinical aspect of the sodium potassium pump, which is very important in clinical practice, that is application of digoxin in treatment of cardiac failure. So, thank you.